Hello and welcome back. After the outline and overview in the previous video, we're now ready to learn more about the processes that are involved in generating ATP during cellular respiration. Without wasting our sweet time, let's have a dive into 5.1.1 glycolysis. Now, I'll be using either glycolysis or glycolysis, depending on how I wish to pronounce the word, so I hope you can get used to it. As usual, let's have a look at the learning outcomes before we dive into the lesson. I hope that by the end of this video, you should be able to illustrate to explain the glycolysis pathway from glucose to pyruvate and also explain and describe the link reaction, which is a conversion of pyruvate to acetyl coenzyme A. Now, to put it simply, glycolysis is breaking down glucose. Memang paling ringkas kalau kita mau faham, glycolysis adalah apabila kita ambil glucose molecule, kita belah dia menjadi separuh supaya dia jadi molekul yang digelar sebagai pyruvate. Tapi cara untuk belah dia tidak semudah yang kita sangka. So in this part, we're going to learn how to define glycolysis, how to explain glycolysis, and also how to draw the process of glycolysis. So let's begin with that definition. You can define glycolysis as the splitting of one molecule of glucose, which is a 6-carbon molecule, into 2 molecules of pyruvate, which is a 3-carbon molecule, through a series of reactions catalyzed by specific enzymes. Glycolysis can occur in the presence or absence of oxygen. Ada ka tiada oksigen, kita tetap akan belah glucose. And this is the first stage in cellular respiration, whether it's aerobic or anaerobic respiration. It will occur in the cytosol, and the free energy released in this process is stored as 2 molecules of ATP and 2 molecules of NADH plus H+. So ini adalah hasil daripada glycolysis. Glycolysis will consist of 10 steps, and each step will be catalyzed by a specific enzyme. We can divide these 10 steps into 2 phases. The first phase is the energy investment phase, so this is from steps 1 to steps 5. We call it the energy investment phase because we will be using ATP during this phase. The second phase is the energy payoff phase, which is from steps 6 to 10. And energy in the form of ATP will be formed during this stage. Okay, so kita akan ngam-ngam belah kepada 2 fasa. Dan apa yang kita akan belajar lepas ni adalah kita akan go through step by step. Macam mana kita mula dengan 6 carbon glucose menjadi 3 carbon pyruvate. For this next phase, I hope you take out a piece of paper, preferably A4 size or whatever size you prefer, and draw each of the steps along with me. So kalau dalam gambar nanti, pada step itu ada 6 biji karbon untuk menunjukkan berapa banyak karbon dalam molekul tersebut. Kamu pun ikutlah lukis berapa biji karbon dan apa enzim yang terlibat untuk step itu. So I hope you'll follow along, and let's have a look. Let's have a look at the energy investment phase. So I hope you would start your diagram with a 6-carbon glucose. In this first step, the enzyme that is involved is hexokinase. In this step, glucose is phosphorylated and it receives a high energy phosphate from ATP to increase its energy level to become glucose 6-phosphate, which is a more reactive compound. So dalam diagram kamu, sila mula dengan 6 biji karbon untuk glucose. Ingat, phosphorylation adalah penambahan phosphate group. So kita ambil satu phosphate group daripada ATP. ATP menjadi ADP. Phosphate group itu kita lekat di hujung. Sekarang dia sudah digelar sebagai glucose 6-phosphate or G6P. That brings us to step number 2. The enzyme involved in step number 2 is phosphoglucose isomerase. In this step, glucose 6-phosphate is rearranged to become fructose 6-phosphate. So di sini, kita akan susun balik structure dia, supaya daripada glucose, dia sekarang digelar sebagai fructose. Tapi bilangan karbon tetap sama. That's why it's called an isomerase. And that brings us to step 3. The enzyme that will be involved in step 3 is phosphofructokinase. You see the word kinase, you can already guess it's going to involve phosphorylation. In step number 3, fructose 6-phosphate is activated by the addition of phosphate from ATP to form fructose 1,6-bisphosphate. So, enzyme phosphofructokinase akan ambil fosfat daripada ATP, lekatkan kepada fructose 6-phosphate. Sekarang dia sudah menjadi fructose 1,6-bisphosphate. Saya lebih suka gunakan bisphosphate, tapi ada juga buku yang akan gunakan diphosphate. Dua-dua merujuk kepada molekul yang ada 2 phosphate group. Tapi perbezaannya, dan kenapa saya lebih suka guna bisphosphate, adalah bisphosphate merujuk kepada molekul di mana 2 phosphate group dia tidak melekat dengan satu sama lain, dan melekat kepada atom yang berbeza. Manakala diphosphate biasanya merujuk kepada 2 phosphate molekul atau phosphate group yang melekat dengan satu sama lain ataupun bersambung kepada atom yang sama. So, although some books write fructose 1,6-diphosphate, I would prefer fructose 1,6-bisphosphate because if you look at the structure, bisphosphate is the more correct way to describe the molecule. I hope you'll be able to remember that, and that brings us to step number 4. The enzyme involved in step number 4 is fructose bisphosphate aldolase. So what we're gonna do in step number 4 is fructose 1,6-bisphosphate is split into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Secara ringkasnya, apa yang berlaku di step nomor 4 adalah kita akan belah dia di tengah-tengah, aldolase, right? So the function of aldolase is to split things without water. So we split it there, and it's going to form glyceraldehyde 3-phosphate, also known as G3P. And the second molecule is dihydroxyacetone phosphate, which is DHAP. Nampak macam dia benda yang sama, cuma terbalik. Tapi ingat, dalam biology, dalam kimia, bila dia punya bentuk berbeza, namanya berbeza. Dan properties dia pun berbeza. Tapi kita tidak perlukan DHAP. Kita perlukan G3P. So what's going to happen next is step number 5. Since we need G3P, we just have to change this into this. The enzyme that's going to involved in that changing is enzyme triose phosphate isomerase. Dihydroxyacetone phosphate is rearranged into another molecule of glyceraldehyde 3-phosphate. Jadi enzyme triose phosphate isomerase ini akan mengubah DHAP kepada G3P. And that is the 5 steps that are involved in the energy investment phase. Now we're going to go on to the second phase, which is the energy payoff phase. In the energy payoff phase, remember here we now have 2 molecules of G3P. So although in the next diagram I show only 1, it's actually going to happen twice. Disebabkan di sini ada 2 molekul G3P. Kedua-dua molekul G3P akan melalui reaction yang sama dalam stage 6 hingga 10. Walaupun diagram saya tunjuk satu saja, tapi actually ada 2 molekul yang melalui benda yang sama. Alright? So keep that in mind as we go on to the next slide. Now let's continue with step 6. Although the diagram here is cut off, I just want to tell you, up here, you can redraw that G3P molecule. Okay, tidak nampak di sini sebab diagram terpotong, tapi sila lukis satu G3P molecule di permulaan. Alright, the enzyme that is involved in step 6 is an enzyme called glyceraldehyde 3-phosphate dehydrogenase. Cara kita ingat, ini adalah G3P dehydrogenase. Dehydrogenase, what's it gonna do? At this step, glyceraldehyde 3-phosphate is oxidized. So H atoms or hydrogen atoms are removed. NAD plus is going to be reduced to become NADH, and it produced 1,3-bisphosphoglycerate. So nama dia dehydrogenase, dia akan keluarkan hydrogen atom. Dan siapa yang terima hydrogen atom itu adalah NAD+. So it's going to receive that hydrogen atom and becomes NADH. A phosphate group is going to come in. And so our G3P now becomes 1,3-bisphosphoglycerate. Kamu akan perasan sesetengah buku ataupun diagram akan tulis 1,3-bisphosphoglycerate. Ada yang akan tulis 1,3-bisphosphoglyceric acid. And it is referring to the same thing. Kalau di hujung dia, eric acid. Okay, dia boleh juga ditukar nama panggilan lain, iaitu serate. So, kalau dia adalah 1,3-bisphosphoglyceric acid, kita boleh juga pendekkan nama dia atau bagi nama panggilan dia yang lain, iaitu 1,3-bisphosphoglycerate. Dua-dua pun betul. Okay, with that out of the way, we're going to take this 1,3-bisphosphoglycerate and go on into step 7. In step 7, the enzyme involved is phosphoglycerate kinase. In this step, one phosphate from 1,3-bisphosphoglycerate will be transferred to ADP to form ATP. It produced 3-phosphoglycerate. So tadi ada satu ini kan, ini kamu nampak di sini lukisan dia. Ada garisan yang merah, meaning that this is a high energy unstable bond. So right after step 6, when we go into step 7, that high energy unstable bond between this phosphate and the molecule, we're going to use that energy from here and put it into an ATP. Itu yang berlaku di step 7. Ini banyak tenaga, kita ambil tenaga itu masukkan ke dalam ATP. So sekarang yang kita tinggal adalah molekul yang bergelar 3-phosphoglycerate. And we go on to step number 8. In step number 8, the enzyme that's involved is phosphoglycerate mutase. In this step, what the enzyme will do is it's going to rearrange 3-phosphoglycerate into 2-phosphoglycerate. So dia akan pindahkan phosphate group itu daripada molekul ketiga kepada molekul kedua. Daripada carbon atom ketiga kepada carbon atom kedua. And that brings us to step number 9. So in step 9, the enzyme that's involved is enolase. So what's going to happen is the removal of water from 2-phosphoglycerate to produce phosphoenol pyruvate. Okay, so bila kita buang satu water molecule, bond di antara phosphate group dengan carbon molecule ini sekarang menjadi bond yang kurang stabil dan penuh dengan tenaga. This stable bond, this stable covalent bond, now becomes a high energy and unstable bond, which is why you notice the line has now become a red squiggly line. So with this high energy, we're going to go on into the 10th step, where we are going to use the enzyme pyruvate kinase, and that phosphate will be transferred to ADP to form ATP. So semua tenaga yang ada pada bond yang tidak stabil ini akan dibawa ke dalam ATP. And now our phosphoenol pyruvate is converted into pyruvate. and that is steps number 6 to 10 in the energy payoff phase of glycolysis. And so far, we have already completed the 10 steps in glycolysis, and we have looked at the energy investment phase where we use 2 ATP, and in the energy payoff phase where we formed 4 ATP. Walaupun diagram tadi kita nampak ada 2 saja, step di mana ada ATP, kamu harus ingat itu energy payoff phase melibatkan 2 molekul G3P. So what you saw in the previous slide will occur twice. That means step number 7, 2 ATP. Step number 10, 2 ATP. Total 4 ATP formed during energy payoff phase. So the whole summary, the products of glycolysis is from 1 molecule of glucose, you are going to get 2 molecules of pyruvate, 2 ATP, and 2 NADH plus H+. And what we had from earlier was just this part of the diagram. Apa yang tadi, glycolysis, cuma bahagian ini pada diagram keseluruhan ataupun pada proses keseluruhan aerobic respiration. So ATP yang terhasil tadi kita boleh terus guna dalam sel kalau sel perlukan, dan NADH plus H plus yang kita hasilkan daripada glycolysis akan dibawa kepada electron transport chain. The NADH that we produce from glycolysis will be useful later, and we're going to learn about electron transport chain and chemiosmosis, oxidative phosphorylation, the whole thing in 5.1.3. So we're done with the green part of this diagram. We're going to go into the purple part now, and that brings us to the next process, which is called link reaction. In link reaction, the pyruvate from glycolysis process will now enter the mitochondria. So it's moving from the cytosol into the mitochondria. In here, the pyruvate is converted into acetyl-CoA, and acetyl-CoA will be used in the Krebs cycle. Pyruvate will undergo oxidative decarboxylation. So let's have a step-by-step look at link reaction. So we're going to start with this pyruvate, which is here in the cytosol. It's going to enter into the mitochondrion via a transport protein. That transport protein will be on the membrane of the mitochondria. And once it enters, we will start with the first step. Bila dia masuk saja, ini adalah step number 1. This process is decarboxylation, where we remove 1 molecule of carbon dioxide. So the product in step number 1 is carbon dioxide. The second step will be oxidation. The product from this step number 2 is acetate. So what's going to happen is the molecule will be oxidized and it's going to lose hydrogen atoms. So siapa yang terima hydrogen tersebut, itu adalah NAD+, dia terima, dia menjadi NADH plus H+. So NAD+ will be reduced into NADH plus H+. And then the third step is the addition of coenzyme A. So kita akan masukkan coenzyme A ke dalam molekul ini dan kita akan dapat acetyl-CoA, which is the final product of link reaction. So it's quite the short and simple process, and we started with a pyruvate, we end with acetyl-CoA, but not just acetyl-CoA, we also produced 2 molecules of NADH plus H+, and 2 molecules of carbon dioxide. Kenapa boleh nampak 2 di sini walaupun di sini nampak 1? Dalam diagram ini adalah hasil untuk 1 molekul pyruvate. Tapi biasanya bila kita cakap pasal products of reaction, kita merujuk kepada berapa biji glukos hasilkan berapa biji product. So if we're talking about how many products can we get from link reaction if we start with 1 molecule of glucose, from 1 molecule of glucose we can produce 2 molecules of acetyl-CoA, 2 molecules of NADH plus H plus, and 2 molecules of carbon dioxide. And that very simple part is explaining the link reaction. Now, during the link reaction, of course, we produce this NADH plus H plus, and those NADH will be brought over to the electron transport chain too. So we'll see what happens to them in the upcoming videos. So that's it for this video, just a very quick summary of what we've gone through in this part of the lesson. We've already looked at glycolysis and the products of glycolysis. Remember to draw the steps. And also, we talked very briefly about what happens in link reaction and the products of link reaction. So what should you do now? You should come up with your own notes and also complete the Chapter 5 worksheet. There is the aerobic respiration worksheet, which you can find in the GC. And you can do it, complete it up until the part about link reaction. That's about it for this video, and I will see you again in the next one as we learn about the Krebs cycle. Thank you, and bye!